I'm Barack. I apologize in advance. I am going to show you pictures of dead stuff in this talk. So if you are sensitive to blood, sorry. Okay, so I am going to talk about food webs a lot today. And I did want to start with just this, which is why am I talking about food webs at a species distribution modeling meeting? And basically, species distribution modeling gives us information about, habitat species relationships. It also gives us a lot of information about co-occurrence. And this is really cool. This is certainly something we want to know. But it doesn't give us information about interaction. And interaction is also very important. Sorry, I told you it was just going to be. Barack made a lot of these slides. And he keeps coming to my one-on-one meetings and being like, look at what you're making me do. And then it's just like a dead baby lion. And I'm like, okay, I'm sorry. No. Anyway, so we're interested in interaction. And so there's kind of this big open question or open debate right now, which is, does co-occurrence imply interaction? And there's work that suggests that yes, it does, right? The fact that things exist together with some other stuff implies that maybe they interact. This is a paper from 2020 that specifically was looking into this. Why is interaction important? Well, you don't just need to know the distribution. If we want to actually understand ecosystem function and diversity, the structure of interactions is really important. I pulled this paper. It came out 2 days ago. I thought it was really cool. They basically were looking very specifically at dung beetle diversity and doing a systematic study over 15 years, exploring the impact on the diversity of dung beetles if you did or did not have elephants in these kind of specific enclosures. And they basically found that if elephants go extinct, the diversity of dung beetles goes down significantly, right? So you wouldn't be able to predict that from co-occurrence alone, right? I mean, maybe if you had enough data. But as things are changing, we really want to understand not just where things are, but how they're actually interacting trophically in abiotic interactions, how they interact with their environment, but also these biotic interactions. I think there's kind of a very classic example that maybe all of us are very familiar with, which is insect populations are declining, right? What does that mean all the way up the food chain, right? What is that going to impact? And can we even understand or start to predict? All right, so hopefully I've convinced you that we do care about what eats what. So how do we actually observe what eats what? So this is I also wanted to put a paper from 100 years ago. So this one is from 1923, and it's the food web from Bear Island. And it's basically this barren spot in the Arctic, and they went in and they just looked at a lot of things. And this is kind of the very classical version of these types of food webs. We would go and witness them in person. You would just go try to watch. And there's a lot of this still going on. I was in Rwanda last year, and I went out with one of these chimpanzee tracking teams. I met a guy who had been following the same group of habituated chimpanzees for 30 years every day, 6 days a week. They have a team, and they go out every single day, and every 15 minutes, they record the behavior of the chimpanzees. They're just watching them all the time. And they're writing down everything they eat as one of the things they're observing. That's crazy to me that there is a person who has spent 30 years of his life watching and recording every 15 minutes, things like this, right? Clearly, this is not very scalable. But also, it's really hard to actually witness these predations in person, right? These predation events can be brief. They're often nocturnal. They can happen underwater, underground, high up in the canopy. It's just really difficult to observe. And that means that direct sightings are rare and often opportunistic. And we certainly have systematic studies like this one, but they're rare and they're really only for some species in some places. So the other way that we observe interactions is through physical evidence. So this is stuff like dissecting the stomach of an animal or going into scat or owl pellets, dissecting that, trying to figure out what's there. Digested or broken remains are pretty hard to identify, so there are some issues with this. Now we have molecular tools, so things like eDNA or DNA metabarcoding of gut or scat or stable isotopes. So there are tools that help us directly query physical evidence, But this does require you to capture specimens, do field work, you need lab time, and you need appropriate reference libraries. And if anyone is like me, very curious about how we actually use these types of DNA processes in practice, I think there's just a lot to still figure out, right? But we can get physical evidence, so we can certainly go and find some of these interactions. But again, it just really doesn't scale. And I think I just want you to like build this mental model of when we think about interactions, how many there are, right? So how many species are there? Okay. How many species are there just in Europe, right? Now you need to do that many times that many again pairs, and maybe we can constrain based on functional traits or something else, but there's a lot of possible interactions. So, this is something people know is important. So there are programs that the Globi is a really great example. It's this global biotic interactions database. And what they've done is they've gone into prior scientific literature and existing image databases, and they've tried to surface evidence of interaction. This is a pretty heroic effort. It is certainly noisy. I've certainly dug in, and there's definitely, reported interactions that are not necessarily that are true or not. There's actually a really fun, if you go to data sets or data, there's one of these where you can actually go and find the refuted interactions, which is basically the whole set of interactions that people are arguing about. And you definitely get these types of arguments when the reported interactions, the evidence of interactions is just someone saying, I saw it once, right? It's very hard to argue with an image that actually shows it. So this is a great start, but there's still a lot of interactions from Globi missing. And in particular, there's a lot of interactions from Globi missing that you would kind of expect to be there. So for example, we don't have any evidence of American pumas eating American wild turkeys. But probably that's happening. It would be very surprising to me if that wasn't happening. And that's big, we were just talking about you're talking about these big trophic, big biomass species. We're missing some interactions that you would kind of expect to have. So maybe iNaturalist to the rescue once again. We've got, 300 million images in iNaturalist. And it turns out there are a lot of images of interactions in iNaturalist. But now the challenge shifts from observing the interactions to finding them in this database. I think this is very promising. Now imagine, I told you about, the n choose n complexity. Try imagine training a supervised model for this problem when we don't know if the images are in there in the first place. So I think pretty clearly this is not going to be a supervision thing. So we've been working for a while, and maybe some of you guys have seen on this type of okay, what can we retrieve with open language? And, we have some papers benchmarking this. We had a recent paper that just got accepted at Ecological Informatics, where we kind of took this system that was just retrieval and actually worked with a bunch of scientists, including Laura, who's great, and a bunch of other people from ABC, and tried to go from just a retrieval system to something that would actually generate scientific evidence. And we have a food web case study in that paper, right? We were able to actually look at not just generating a food web, but actually look at trends in how diet is changing across seasons for a specific subset of species. This was led mostly by Marta Jarzina and her students. So to me, and this is important because we always talk about evaluation, here, the red is the trends that we estimate using search and kind of verification. And the blue is the estimates that they got from a previous effort to curate these types of seasonal diet variations from the literature. So they match up pretty well, better than I expected, honestly. So to me, this is convincing that there is useful interaction and particularly trophic food interaction information in iNaturalist. So now I have a postdoc, Barak Hakim, we're looking at, okay, how do we fill in the blanks in the food web as efficiently as possible? I want to go get as much information as we can out of iNaturalist as possible to just fill in the blanks. So now the question is a bit different. It's not just do we have a retrieval system? The question is, what interaction should we prioritize? And we were working a lot with Laura on trying to think about what is ecologically important? How do you kind of rank the importance of edges on this graph? And then we also need to figure out what interactions we're likely to find. So, what's likely to be an iNaturalist? what are we likely to find with the VLMs that we have? And what are we likely to be able to verify? Because it turns out you get a lot of things eating miscellaneous meat. And it's hard to figure out exactly what that miscellaneous meat is. And so in terms of what interactions should we prioritize, this is still very much ongoing work. But kind of one way of thinking about this is looking at metawebs, like basically taking from ecological theory, and which metawebs are basically this combination of theory, species traits, and the sparse existing evidence from the databases that we have to try to directly estimate the full web. So I thought this was fun. This came out last year, a species level multi-trophic metaweb for Switzerland, appropriate for this conference. But yeah, they basically have these checklists of co-occurrence. They have empirical interaction data for a very, very small subset. And then they're assembling the metaweb based on some information about functional traits. And then they can actually try to infer, these other interactions. So that's a start. And we're still iterating on exactly how we prioritize from the ecological side. But then if you're trying to get at what interactions are we likely to find, this is also a mess, right? So if it's in iNaturalist, that means someone has to go to the place, which means it has to maybe be accessible. You have to notice the interaction, which means it has to be visible. You have to have taken a photo of the interaction, which means maybe it has to be charismatic or something. You have to upload it to the internet, hopefully in iNaturalist. And then our VLM has to be able to find it. So then we have to, have good search performance for some very fine-grained stuff. So the first bit is something we're working on estimating. Like, how do you actually build kind of an estimate of findability, but also an estimate of likelihood to be in there? So you're just building these kind of active estimations of what is in iNaturalist. But we also just tried to get started on just probing the sensitivity of these VLMs relative to these prompts for fine-grained species interactions. So we have a very, very preliminary set of data just to start doing experiments where for every A-B pair, we collected an image of the predator eating the prey, an image of just the predator, an image of just the prey, an image of the predator eating a different prey, an image of the prey being eaten by a different predator, and both being present in the image, but no actual predation. And then we randomly sampled iNaturalist data for each of these and verified them. So you get stuff like this, right? So this is like, okay, just the crocodile, just the blue wildebeest, the actual predation event of the two, the co-occurrence, but actually no predation, but are they hanging out? And then, yeah, this is eating a baby hippo. Now, this is a hyena eating the wildebeest. Yeah.
Just to clarify, you're using interactions, entropic interactions.
Yeah. I'm playing it so fast and loose with terminology. I apologize. I mean, specifically predation events.
Okay.
Yeah. I should be more precise.
So if it's predation, then if species, there's predation happening in the event, there's no edges linking the species.
It's certainly not a negative, but it's not a confirmed positive, okay. Right?
Do you feel put an edge on the graph?
No. And we're not. This is not building the graph. Okay. This is actually literally just to try to figure out what the VLM is doing, right? So this is estimating VLM performance. Yeah.
Do you have trends in what stacks up after verification? Like how?
I'll get there.
What they hear.
Okay, so other examples, right? Lion, plain zebra, or sharp-skinned hawk and European starling, or cheetah and Thompson's gazelle. So we have these kind of hand-curated examples of the actual thing we want to find, and then a bunch of hard positives, basically, or hard negatives, sorry. So turns out, just on some simple experiments here, we can find predation, but it's pretty hard to be precise about exactly what's being eaten with these VLMs, right? So it finds predation versus random photos pretty well, but telling the right play from something similar, maybe almost like a coin flip. And this is actually also similar. Laura's had ecology students who have been going looking through the data, and they've similarly really struggled to figure out what is being eaten. Also, interestingly, if we just look at the similarity scores, we get the best similarity scores using the VLM for the co-occurrence images. My guess is that the co-occurrence images, it's just actually easier to see both things, right? One of them isn't mangled meat. So yeah, so this is a complexity. So if you're actually just looking at the, clip style VLM similarity. I think this one is just SIGLIP or something. You end up with much tighter similarity for both present, but not necessarily, or specifically not predation. You can fix this with prompt engineering, basically. Like you can just be clever about some relational set of prompts. We had a decomposing visual search paper at a CVPR workshop last year, and this is kind of just building on that. So you can kind of hopefully build some sort of relation-aware reward for our active sampler without needing to fine-tune the VLM, which I don't want to do because you just lose capabilities super fast in all of our experiences. And then retrieval is biased. Are we even surprised? This is maybe related to what you were asking. It's like, I think it's more just like, if it's easier to see the thing in the image, you're more likely to be able to return it. And I think you're also more likely to be able to verify it.
When you're looking at the species identification, you're not using a separate fine-grained classifier for that.
So we're playing around a bit with both. What I found so far is basically like the fine-grained classifiers are pretty good with the predators, but I think the prey are it's just out of distribution. Like we don't have as many examples in those fine-grained classifiers of like the thing as it's being eaten, which tends to look pretty out of distribution. So they're not as good. And then we also are, as much as possible, leveraging the labels in iNaturalist, right? Because we don't actually necessarily need to categorize everything.
Both label.
No, certainly. You get one or the other, basically. But like you can say it should have maybe one or the other. Yeah. So also Tim Hawk was supposed to be here and couldn't come very last minute. So I did just want to flag that he's been doing some cool stuff with occupancy modeling and deep learning, leveraging ground level image features. So take this as Tim Hawk's poster, but you should go read his paper. I think it's very nice. That's all. We're just getting started on this. I think it's exciting. I did want to flag that Barak's been really like leading the effort for us, but we're also working with Laura and some of her students who are amazing and certainly know way more about food webs than I will ever know. So cool.
No? We have any question from the audience? Maybe you.
Yeah.
I always think that including interactions in like the STM space has a lot of promise, but it seems like your model is also really good at just getting co-occurrence, right?
Yeah.
Because you said
Verified co-occurrence. Verified co-occurrence. Yeah.
Yeah. Because that's usually the only have to label for one or the other.
Or you know that like they were like kind of maybe in the same place over time, but you don't necessarily have this like they're really in the same place.
Yeah.
Yeah.
Because you said at the start of your talk, like SDMs can give us information about co-occurrence, but actually this is something we kind of struggle with, right? Because of lots of things that Alex mentioned in the earlier talk. So I'm just wondering if you've also considered using this not like in parallel, not just for the interactions, but also for co-occurrence.
You can focus on one thing at a time. So we're webs, but if you want to look for co-occurrence using these tools, we have a bunch of stuff set up. I think it's a great idea, and it seems like it might work a little better.
Okay.
Yeah.
In the same vein, interactions that are, I think, and we've
Not predation. Like non-predation interactions.
Non-predation, like pollination.
Yeah.
We found with Inquire actually, it works super well for pollination.
Yes. And I think there's a lot of people working on pollinators. So I think that's really promising. But yeah, no, I think what actually gets really messy though, I don't know. It works super well for plant, like for bug on plant. But I've been having a bunch of discussions with people who study pollination who are sort of like there's just because the bug is on the plant doesn't necessarily mean that it's pollinated. So then they're trying to decide like, oh, can you see the pollen on its legs or like, I don't know. So I don't know, Tanya, if you have had those discussions of like, if it's enough.
Yeah, sometimes bug eats plant, yes.
Mm.
Yeah, other questions?
Yeah.
Wondering whether you could generate a query image.
I have been trying to get generated images to be useful for me for 5 years. And I'm getting really frustrated. Basically, that's where I'm at with that. You can totally generate images that look plausible, kind of, but the morphological accuracy is really complicated. And then like, even if you have human verified, yes, this is the correct morphology for this rare insect or whatever, it doesn't help in your training in a way that is very annoying. Copy paste seems to be pretty good. So maybe I could just do some fancy copy paste stuff.
Also, is it something desirable? Because I can imagine that now we have more and more deepfakes, and usually for the picture was an evidence. So when I saw plenty of videos, Black Lion attacking like tourists and like not really true, right? So there's something actually.
Yeah, do I need to start working with
Yeah.
fake detection for this to be valid? Yeah. Yeah, I think this is fair. I think at least for now, I'm just gonna hope that most of the stuff on iNet is real pictures, but I actually know, like I talked to Scott a bunch and they are really struggling with this, particularly because what you have, I'm very empathetic to this. You go out, you see something very cool. You try to take a picture with your phone. It looks like crap. And then you're like, people just are like, what if I just generated the photo I saw, And so they're ending up with like kind of well-intentioned, but like generated images in iNaturalist, which is really difficult because then is it actually correct? Yeah.
So we will have to start developing deepfake algorithms. Oh, I thought we were in a very nice field because data were sacred.
Yeah, but so did you guys see there was like really splashy online? There was some like Japanese conservation land, like protected area, and they had a camera trap image that didn't look that nice of like something eating something. They were really excited about it, and so they like used generative AI to like make it look cooler. And then people got really upset, notably, because they're like, oh no, you're like corrupting evidence. This is like misinformation. Anyway, so happening. Yeah.
Yeah, so one of the things we've been experimenting recently is searching embedding spaces of models like Bioclip.
Mm-hmm.
And so you can do a prompt, right, and see what's wrong. And I've been just experimenting with this, with lion eating different things. And even before that, like you can actually get, depending on what the focus of the image is, you can get like with a prompt of a lion eating, visual prompt of a lion eating something, you'll get a lot of stuff that lions might eat.
Mm.
And then you can flip it of like, let's say a zebra being eaten by something, or a zebra is in the foreground, and you'll get a lot of stuff that eats zebra. And so you can get these kind of directed search.
But that's almost like it's a prior, right? Like so I guess we're pulling our priors right now from the Metaweb. So it's like, I think the priors are like coming from ecological theory, but I wonder if there's actually very different. It would be interesting to see if like that Bioclip prior would match or would not match kind of like the Metaweb prior on food.
Yeah, that was essentially the question, because nothing in Bioclip has any priors on interactions.
Yeah, good question. I don't know. I think we're going to stick with the Metawebs for now because we already set it up, and the theory is pretty solid. But I think that's really interesting to try to understand. Maybe that's more of an interpretability thing. Like, what is like the priors coming from Bioclip? Like, are they different or the same as like? And then if you look at the differences, like maybe there's something interesting there.
We experimented a little bit more with pollinators,
Mm-hmm.
and pulled out full lists of what does this moth pollinate, right? So
Cool.
by just visual prompting. And so I'm wondering if it can, like, particularly filling out these A eats B, A eats C, A eats D, like all of these.
Yeah. I think my question is just like, is that the same as what the kind of theoretical expectation is, or is it different? And when it's different, what does that imply, right?
Yeah, it also expands a little bit your data search.
Yeah. I mean, a lot of this work is actually kind of this like, what we're really trying to do is figure out like, how do you direct human effort in a search process? And so a lot of it is like building these kind of like active samplers that help us like direct the search effort to try to actually discover evidence as fast as possible. And for me, at least, like the specific question isn't as important as like the process of trying to speed up search. But I really like that idea. I'll talk to you about it. That sounds cool. All right, I should go.
Okay, I think that we should move on.